Assalamualaikum warahmatullahi wabarakatuh, and a very good afternoon to all the audience. First of all, I would like to thank the organizing committee for inviting me to give a talk in the 19th annual scientific meeting for the College of Pathologists, Academy of Medicine, Malaysia. I am Shahrun Shamsuddin. I'm a lecturer from the Biomedical Science Program, School of Health Sciences, University of Science Malaysia, Health Campus in Kota Baru, Kelantan. I will share my slide. Okay, this is actually the work that has been done by a group of my students who actually trapped during the MCO last year in the hostel. They cannot go anywhere, so they just sit down and do a lot of readings. And then one of the important interesting part that they have produced is to look into the molecular evolution of the SARS-CoV-2 originated from Wuhan until now, the Omicron, BA1, BA2, and also all the recombinant subvariants. Before we go further into the history of the emergence of this virus, let's look into the structure of SARS-CoV-2. As we all know that SARS-CoV-2 is actually a single-stranded RNA virus, which consists of 4 important outer layer protein, which is actually decide on the infectivity on human infections. Okay, and the first protein, the most important one, is the spike protein, which is actually the protein that is involved in the interaction with the ACE2 receptor in the surface on the surface of human cells. And then we have nucleocapsid protein, which is actually a cover protein for the virus. And we have also membrane protein, the M protein, and also envelope protein, E. Okay. If we look the genome size of human coronavirus is about 27 to 32 kilobase pair, and the size of the virus is about 100 nanometer up to 160 nanometer, depends on the variants. So if we look into the genome of the virus, you have the structural genes, which consists of the spike protein, the envelope, the membrane, and also the nucleocapsid, and also the non-structural gene, which is actually PP1A and PP1B. And this non-structural gene is actually very important for the production of transcriptional and translational cascade for the purpose of propagation and also replication of the virus. So once the virus infect into the human cells by using the binding of spike protein to the ACE2 receptor binding, it will allow the whole virus to go inside the cells, and then there will be a somewhat long process where all the genetic material of the virus will come out, and we'll carry out all the transcription and also the translation. And you can see here the protein of PP1A and PP1B. They will produce a lot of protein, which is actually very important to control the transcriptional and also the replication of the virus in the cell. Okay, and for the human, the most target for this virus to infect is actually the respiratory airways and also the eye. Okay, this is the two target organ that normally SARS-CoV-2 can go and interact. if we look into the emergence of this human coronavirus before 2000, after 2000, the coronavirus becoming severe. Before that, it was very mild, and the first human coronavirus reported is in 1966 in Chicago, in the United States, and also in Maryland in 1967. And scientists was suggesting that the same variant was suspected to be the cause of the Russian flu that happens in the 1889 in the 1800, where it had killed almost 1 million people. So this is the first and second reported human coronavirus, and then most of the patients that interacted with human coronavirus, they are very exposed to animal. So scientists had a postulation that maybe that the virus is actually originated from animal, and then they use the animal as a reservoir, and then when it comes to the time, they will infect human. Okay, and the most animal that has been reported related to the as a reservoir for human coronavirus is bats, the horseshoe bat, genus Rhinolopus. Okay, this is the bats, and other than that, you have civet, camel, pangolins, camelid, and also rats. Latest data describing that cat also might be harboring, as the place where human coronavirus can go and in fact becoming the reservoir. Okay, you can see here that the bat is mostly the culprit behind as a reservoir, where the SARS-CoV-2, SARS-CoV, the MERS-CoV, okay, originated. And after 2000, after the year 2000, there are some sort of emergence of severe strain of human coronavirus that they call SARS. This is actually known as severe acute respiratory syndrome, which is also known as atypical pneumonia. It was first well documented human coronavirus that caused pandemic in human history, and the agent is SARS-CoV. Okay, SARS-CoV. In this case, now we are calling it the first SARS-CoV, because COVID-19 is SARS-CoV-2. It was firstly detected in Guangdong during the middle of November 2002, and in February 2003, a few months after that, the SARS epidemic started to spread to Southeast Asia, and it becoming nationwide outbreak in March 2003. Okay, and WHO named this disease as a pneumonia SARS. Okay, and then in April 2003, the epidemic growing rapidly. However, in May 2003, the number of the outbreaks is getting less and lesser. It began to decrease. And in June 24th, 2003, the World Health Organization announced that the epidemic in mainland China for SARS-CoV that time has been extinguished. So the epidemic is somewhat like not even 2 years. Started in November 16, 2002, and end up in June 24th, 2003. So this is the first emergence of severe strain of human coronavirus, the one that is known as SARS. Okay, after 2000, there are somewhat like a bubble outbreak with this human coronavirus. In 2004, there are 2 cases of SARS-CoV both were detected in the Netherlands. Okay, and then in the same years, 2004, another strain variant was detected in Hong Kong. However, all these variants they are not severe. Most of them cause very mild respiratory disease, and they did not mutate and did not becoming pathogenic to human. It's only in 2012 when the first Middle East respiratory syndrome that they call it, when one patient has developed acute pneumonia and renal failure in Saudi Arabia. So they announced that WHO has announced that the new human coronavirus, the severe new human coronavirus, start coming out again. They call it MERS-CoV. Okay, and the main difference between SARS-CoV and MERS-CoV is that the patient of MERS-CoV developed acute renal failure, which is very unique for MERS compared to the SARS-CoV. Okay, and the last one is the one that we are calling COVID-19. In late December 2019, the SARS-CoV-2 virus has emerged again, firstly detected in Wuhan, China, on the 7th of December 2019, and then the whole gene was characterized in January 2020, and then on 24th of January, they have announced that they have evidence to suggest that SARS-CoV-2 is actually originated from the wildlife. Okay, and WHO has declared the outbreak as a public health emergency of international concern on the 30th of January 2020. And then the rest of the things is already history. We all know. It came to Malaysia. It came out with so many things, many stories. And this is actually just to show the comparison of the main severe human coronavirus. You have SARS-CoV, which is emerged in early 2000 in Guangdong, China, and you have MERS-CoV, which is originated, that came out, emerged in Jeddah, Kingdom of Saudi Arabia, and then the last one, the one that has been known as COVID-19, the SARS-CoV-2, that has been firstly detected in Wuhan. That's why the topic is Wuhan. Okay, so you can see here, I just took some details about the different of the symptoms of each human coronavirus on human. Okay, just looking into the classification of the viruses, just to show where this coronavirus is located in the world of virus. You can see that coronavirus is actually is an RNA single-stranded positive envelope virus, which is actually next to orthomyxoviruses. This is actually orthomyxoviruses, the main genus of class of influenza. Okay, influenza virus. That's why some scientists they call SARS-CoV is basically like a cousin to the influenza. Even when the scientists studying SARS-CoV-2, when they sequence the genes, they park the genes, the genome, at the GSAID, which is actually originally as a gene bank for influenza viruses genomes. Okay, and the viruses was actually coming from beta coronaviruses. Okay, the MERS-CoV, SARS-CoV, and also SARS-CoV-2 is actually has been classified as a beta coronavirus. Yeah? And comparing in terms of genome similarity. SARS-CoV-2 are having 80% similarity with SARS-CoV, 50% similarity with MERS-CoV. Okay, but SARS-CoV-2 is the highest transmittable variant compared to SARS-CoV and MERS-CoV. Okay, and if you see here, this is the SARS-CoV-1 interaction of ACE2 receptor towards the spike protein of the virus. And then for the SARS-CoV-2, you can see with the mutation that happens throughout the evolution, it produces the interaction even better. It binds more tightly towards the receptor that makes SARS-CoV-2 becoming more fatal and also more transmittable compared to the previous strain variants. Okay, so you can see starting from December 2019, the Wuhan strain, how it evolved with mutations to become D614G variants in 2020. It's about a year. Okay, what has happened was that the aspartic acid on the position of 614 amino acid changed to become glycines. That's why the variant is called D614G. Okay, and it took about a year. Okay, and in May 2020, WHO has came out with a new way of labeling, like easy to say label for both for all the variants of this SARS-CoV-2. The first one is the variants of concern, and also the second one is the variants of interest. And the variants of concern is actually the list of variants that are severe, that put people in hospital, and also showing irregularities and also hard to be detected. Okay. And then they have a few that time there were like 5 or 6 variants of this VOC. The first one is Alpha, which is originated in England, in UK, and the second one is Beta, which is originated from South Africa, and then Gamma from Brazil, and also Delta that originated from India. If you still remember at that time in the middle of 2020, people were shocked with the fatality, severely fatal Delta variants in India. And then you have the latest one, okay, the one that is now dominant in the world now, the Omicron. Okay, the Omicron. And this Omicron, they have a few sub-lineage. They have BA1, BA2, BA3, BA4, and so on and so forth. And also the recombinants of Omicron, because all these BA1, BA2, BA3, BA4, they can interact each other to do recombination and produce what they call the new variant of XE. Okay, the new variant of recombinant Omicron. This is actually just to show the difference of symptoms between Omicron and Delta. Okay, you can see that in most cases, the symptoms of Omicron are barely the same like the Delta one, except the loss of smell. Okay, the loss of smell here, you can see that Delta variants are showing a very different loss of smell capability to the patient compared to the Delta. And this one also, the altered smell. Okay. Other than that, shortness of breath, sneezing, sore throat, very the same. Okay. And the Omicron has taken over the world dominancy starting from late last year, November, and in Asia it took like December. Now they are dominant in Asia. Okay, And you can see here the reason why Omicron is becoming easily to become dominant is because they mutated a lot, almost 30 sites of mutations, okay, for the Omicron, and they produce 3 lineage here. You can see compared to the green one, which is the Delta. You can see less than 15 mutation sites for the Delta and the others as well. So so this is basically just to show how the variants of the SARS-CoV-2 coronavirus evolve based on mutations. Okay. Now let's look into the Omicron XC, which is the recombinant. This is actually the recombination of different lineage of Omicron that produce new recombinants. Okay, they have reported 1,179 cases of Omicron XC in the United Kingdom, and the current evidence suggests that there is little cause for concern. Luckily, it is not as severe as they used to have before. Okay. However, scientists and the doctors are still monitoring this sublineage. Hopefully that it will not evolve into the worst kind of variants. Okay, so this is actually just to show how the recombinant occur in the cells where 2 different Omicron get together, and then there will be a recombination. Simplified by this, you can see the mixture of the green and the purple color, how they go inside encapsulation into the components of virus, and then after that being released into the environment of the human cells. Okay. So the symptom of the Omicron XC is basically just like the same Omicron: shortness of breath, fatigue, some pain at the body, and then also high temperature. You have fever, and so on and so forth. So this is basically the same like what we have from the normal Omicron. I think that's about all. Just a very small sharing information regarding the evolution of this SARS-CoV-2. I would like to say thank you again to the organizing committee, and Assalamualaikum warahmatullahi ta'ala wabarakatuh. I will stop sharing. I will stop that.